So chapter 12 talks about gene expression and synthesis of proteins. So we're going to be talking about DNA replication, transcription, and translation. So DNA replication occurs in the nucleus. Transcription also occurs in the nucleus, but translation occurs in the cytoplasm. So know that key difference. Again, we've seen this picture before. In a prokaryotic cell, there's no nucleus, so the DNA is sort of everywhere. But we know in the eukaryotic cell, since we do have a nucleus, that's where your DNA is going to be located. So here are your learning objectives. Be sure you can answer all of these completely. So your DNA structure is made of three main things. First thing is the 5-carbon sugar, so deoxyribose, since we're talking about DNA. A nitrogen-containing base, so those are your purines and pyrimidines, and then a phosphate group. So you should know the three things that make up a DNA structure: 5-carbon sugar, nitrogen-containing base, and a phosphate group. So if you look at the picture, you'll see that your DNA structure is made of two strands of nucleotides. So the bases are going to pair, and that's called base pairing, by hydrogen bonds. So using X-ray diffraction, they were able to determine that DNA is actually a double helix. And this double helix structure is extremely stable, so it protects all those nucleotide bases from attacks from water-soluble compounds and water itself. And then it also provides an easy way for replication. So here we have a picture of RNA. So since we're using RNA, we have to use the sugar ribose and not deoxyribose. But the concepts are the same. So you need 3 units to make RNA. You need your sugar, which is ribose. You need your nitrogenous base, and then you need your phosphate group. So those 3 together make a nucleotide. And if we want to join one nucleotide to another nucleotide, then we have to use something called a phosphodiester bond. So here are your nitrogen bases. You have your purines and your pyrimidines. So your purines are adenine and guanine, and your pyrimidines are cytosine and thymine. So you should know which one are your purines, which one are your pyrimidines, and you should also know what base pairs with what. So adenine is going to base pair with thymine, and guanine is going to base pair with cytosine. So memorize these structures. They're going to be important for the tests. Notice that purines have 2 ring structures and pyrimidines have a single ring structure. So this is a picture of deoxyribose. So you need to know the difference between the sugars deoxyribose and ribose. So if you look at the second carbon, in DNA, it's going to have a hydrogen in that second position. So that's why we call it deoxyribose, because it doesn't have an oxygen. For RNA, at that second position, it's going to have another hydroxyl group there. So this is a new slide. It wasn't in our lecture, but it is a learning objective. It's learning objective number 3. Know the numbering system of atoms in DNA and RNA. So you may want to ask him what you need to know from this slide, but it looks like you may have to memorize the numbers of certain atoms. So here we have nucleoside and nucleotide structures. So you should know a nucleoside is just your sugar and the base. A nucleotide is the sugar, base, and a phosphate group. So if it's one phosphate group, it's going to be a nucleoside monophosphate. If it's 2 phosphate groups, it's going to be a nucleoside diphosphate. And if it's 3 phosphate groups, it's going to be a nucleoside triphosphate. So here we have a table with the bases and then their corresponding nucleoside names. So again, remember nucleosides are just the sugar and the base. In the table, it's shown with using RNA as the sugar. So adenine is the base, the nucleoside is adenosine. Guanine is the base, the nucleoside is guanosine. But if it was DNA, then you would just put the word deoxy in front of it. So adenine would be deoxyadenosine and guanine would be deoxyguanosine. So just be able to differentiate between the names and if it's DNA or RNA. So earlier we saw a polynucleotide of RNA. Here is a polynucleotide of DNA. So they look very similar. The only difference is the sugar. So the sugar for DNA is deoxyribose, whereas the sugar for RNA is just ribose. So we're starting it from the 5' end. DNA is always read from 5' to 3'. Remember, we have 3 units that make up DNA. So we have our phosphate group, our deoxyribose sugar, and then our nitrogen base. So if I want to make nucleotides join together, then I have to use a phosphodiester bond. Here we have a base pair. Thymine is your pyrimidine, adenine is your purine. They're base pairing using hydrogen bonds, and then Dr. Schroeder wants you to know the atom and number that each base forms with the sugar. So if you look at the N1 of thymine, that goes to the chain, and then the N9 of adenine goes to the chain. So those would be the atom and their numbers that form that glycosidic bond to the C1 of the ribose sugar. So here's the other base pair. Cytosine is your pyrimidine, guanine is your purine. The numbers aren't shown here, but it's the same. So the N1 of cytosine is going to form that glycosidic bond with the sugar, and then the N9 of guanine is going to form that same glycosidic bond with the sugar. So if you look at the DNA helix, you'll notice that both strands are anti-parallel to each other. So that means they run in the opposite directions. So on one strand, it's running from the 5' to 3' direction, and on the other strand, it's running from the 3' to 5' direction. So make sure you understand that concept, because you're going to need it to understand the mechanisms of replication and transcription later on. Also take a look at the base pairs adenine and thymine. So you'll notice that they form 2 hydrogen bonds, whereas guanine and cytosine form 3 hydrogen bonds. So that bond's a lot stronger. So here we have uracil and thymine. Uracil is present in RNA and thymine is present in DNA. So if you ask the question, which base are we going to use in DNA, it would be thymine, and if it was which base we use in RNA, it would be uracil. So notice the difference in their structure. The difference is the methyl group. Again, if we're talking about RNA, adenine is going to base pair with uracil, not thymine. So adenine only base pairs with thymine in DNA, but everything else is the same. So here we have DNA and RNA again. We've saw these structures before. If we want to join a nucleotide to another nucleotide, we need a phosphodiester bond. So here, take a look at the shaded blue box. That's a phosphodiester linkage, and notice that it runs in the 3' to 5' direction. So just be sure you know that phosphodiester linkages, which are responsible for linking successive nucleotide units, is running in the 3' to 5' direction. So here's learning objective 4, knowing the nomenclature of nucleic acids. It's also a table, so be sure to pay attention to this. So if you look, adenine, nucleoside's name is going to be adenosine. So remember, nucleoside is just the sugar and the base. So if it was DNA, it's deoxyadenosine, and if it's RNA, it's going to be adenosine. And I guess he blocked off the 3rd column, so I guess you don't need to know that, but just familiarize yourself with the nucleoside in DNA and then the nucleoside in RNA. So here's just more information on the double helix. We already know that base pairs form by hydrogen bonding. We know that it's a phosphate backbone because remember, phosphodiester bonds are what link the nucleotides together. And then as the double helix starts twisting, you're going to form a minor group, which is small, and a larger one, which is called the major group. So if you look from the bottom of the helix, and if the helix is spiraling away from you in a clockwise direction, that's called a right-handed helix. So that's A form and B form. If you look at the bottom of the helix and the helix spirals counterclockwise from you, then it's called a left-handed helix or a Z form helix. So when we talk about DNA, there are 3 types of forms. So we have the Z DNA, which is left-handed, the B DNA, which is right-handed, and then we have the aDNA, which is also right-handed. So the B form is the most common one that's found in most of our cells. A form is not found in cells. And then zDNA is linked to transcription. This is just a table comparing the 3 forms of DNA. It's also your learning objective 5, so memorize this table. He can really ask anything about it. For example, no, A form is a right-handed helix with 11 base pairs per turn and 2.6 angstroms between base pairs. So this is still going off learning objective 5, comparing RNA forms and DNA forms. So typically, RNA is known to form an A form helix, while DNA usually forms that B form helix. So that's the key thing. A form usually forms in RNA, B form usually forms in DNA. So this is another new slide that we've never had. You may want to ask Dr. Schroeder what he wants you to know from this, but he might ask you, where can I find the major grooves, where can I find the minor grooves? So knowing adenine is N6 amino and N7, that's where it forms the major groove, or adenine N3 is what forms a minor groove. So just sort of go through these, but ask Dr. Schroeder because it is a lot to memorize. So here's another one of your learning objectives. Learning objective number 6, know the role of the major and minor groove of A form RNA and B form DNA. So here just lists a lot of differences. So most RNA are A forms, as we said earlier, and B form is most common in DNA. So just read through these to know the differences between the two. So this just shows something called hybridization. So you have a strand of DNA, and it's going to base pair with a strand of RNA. So normally DNA pairs with another strand of DNA, but in this case, it's going to form some sort of hybrid with RNA. So here we have learning objective number 8, knowing the effect of alkali on DNA and RNA. So alkali is just anything with hydroxide ions, right? So DNA strands stay intact, but they do separate. So if you look at the top picture, we have 2 strands of DNA. You remember those 2 strands are attached to each other by hydrogen bonds. But what keeps a single strand together are the phosphodiester bonds. So alkali can separate the strands, so breaking those hydrogen bonds, but it doesn't break the phosphodiester bonds. For RNA, strands are degraded to nucleotides. So here we just see that DNA strands act as templates. So when we go through DNA replication, you have a parental strand. Those parental strands are going to separate so that they can act as a template for a new strand to be synthesized. So during replication, strands of the helix separate in a localized region, and then each parental strand serves as a template for new synthesis of DNA. So this is just a picture of chromatin. So what is chromatin? You should know that chromatin is made up of DNA, histones, and other proteins. So simple test question you can ask is, what makes chromatin? So DNA, histones, and other proteins. So here we have a picture of a polynucleosome. All a nucleosome is DNA and histones. So histones, they're highly alkaline. There's 4 core histones, H2A, H2B, H3, and H4. And the purpose of all this is to compact the DNA in the cell. Since DNA is so large, it has to go through special packaging so that it can reside within the cell. And then also in the middle portion, there's something called the solenoid. So that's just the part where further compaction takes place. So this is a picture of the human chromosomes from a male diploid cell. So the male contains 22 pairs of autosomal chromosomes, and then they contain 2 sex chromosomes. So 1 X chromosome and 1 Y chromosome. If it was a female, they would have 22 pairs of autosomal chromosomes, but then they would have 2 X chromosomes and no Y chromosome. So this talks about trisomy 21, also known as Down syndrome. So this is just a chromosomal defect in chromosome number 21. So you have an extra copy of that 21st chromosome, which leads to Down syndrome. So here's a picture of homologous chromosome and the protein products they produce. So each gene on a chromosome is matched by an alternative version of the gene at the same genetic locus on the homologous chromosome. So these alternative versions are called alleles. So we have two alleles of a gene, one from our mother and one from our father. So if alleles are identical, then we are homozygous for this gene, and if alleles are different, then we are heterozygous for this gene. So this is just a picture of eukaryotic mRNA. It has a 5' cap, a poly A tail. It's got start regions and stop regions, and you're going to learn more about this later, so not too much to focus on right now. So here's another slide. It's not a learning objective. I don't know why he put this in, but you guys will learn about this later. So this compares the prokaryotes and the eukaryotes ribosomes. So prokaryotes have a 70S ribosome made from a 50S subunit and a 30S subunit. And then eukaryotes have an 80S ribosome made from 60S subunits and 40S subunits. So that's the main point, but you're going to learn more about this later. So this is a picture of a hairpin, and a hairpin is just a palindromic DNA or RNA sequence that forms an alternative structure by intrastrand base pairing. So it's base pairing with the bases that are on the same strand. So this only occurs when you have a single DNA or RNA strand. This is a picture of a cruciform, so it's pretty much the same as a hairpin, except cruciform uses two strands. So this is just a picture of a DNA structure that contains 3 DNA strands, so a triple helix instead of a double helix, and they say that these are being developed for therapeutic purposes. So here are some secondary structures. We have a bulge. So a bulge is just having a base without a base pair. You have an internal loop, so that's just a loop that happens within the strand. We have the hairpin. Remember, that's the intrastrand base pairing. So this is a unique secondary structure of RNA. Remember, with RNA, they form an A-form right-handed helix. This is a hairpin double helix. So that's a single strand, but since hairpins use intrastrand base pairing, it becomes a double helix. This is the typical cloverleaf structure of tRNA. We use tRNA for translation, which you'll learn about later. So this is a picture again of the tRNA cloverleaf. We have certain bases or modified bases that attach to it, and you're going to learn about this in translation, but for now he wants you to memorize the structures of all the RNA and DNA bases. So this is just another picture, nothing to know from here, but it's just the 3D folding of tRNA. Alright, so the next 3 slides you can skip. They're not learning objectives and they're really confusing. So wouldn't waste your time looking at these. So this is finally getting back to the learning objective. So learning objective 10 is understanding the various spectroscopic properties of nucleic acids. So this is showing the absorption spectra of the common nucleotides. So the key point from here is just knowing that the absorption peak is at 260 nanometers. This is just a picture showing that DNA can be denatured and it can also be annealed. So that's called reversible denaturation. So at the top we have a double helix. If we denature it, then we're going to break apart the strands, and if we anneal it, we're going to put it back together. So it's reversible. So this is just showing heat denaturation of different DNA strands. So heat denaturation depends on pH, ionic strength, size, and the base. So earlier we talked about GC base pairs, right? They have 3 hydrogen bonds, so they have a higher melting point. A and T have only 2 hydrogen bonds, so they have a lower melting point. So that's a key point. So this isn't anything that he'll test on, but it's just showing the effect of G and C content on heat denaturation of DNA. This shows the proton chemical shifts. I think he did test on something from this slide, but I don't remember particularly what it was. You may want to ask him about this slide. Also pay attention to the bottom right. Methyl is 1 to 3 parts per million. Here's another slide that you probably want to ask Dr. Schroeder about. I'm not sure what he would test from this slide or what he expects you to know. So this slide is Objective 11, your clinical correlation, knowing the different types of viruses containing both DNA and RNA, plus examples of each. So DNA viruses possess double-stranded DNA, are adenovirus, smallpox virus, herpes simplex virus, HPV, and Epstein-Barr virus. So viruses that involve single-stranded DNA could be tortinovirus and paravirus B19. For RNA viruses that possess double-stranded RNA genomes are going to be rotavirus and reovirus. Viruses that possess positive-sense single-stranded RNA genomes are going to be your picornavirus and then some other ones he lists. And then viruses that possess negative-sense single-stranded RNA is going to be Ebola. So this slide talks about the last objective, Objective 12, know some of the antiviral drugs against DNA viruses. So on the right are all the types of drugs that we can use for certain viruses of DNA. So just follow the color coordination. A through B in the purple are going to be for HPV. The green boxes are going to be for HSV. Red boxes are going to be for cytomegalovirus. And then your blue box at the bottom right is going to be for your varicella zoster. So he didn't test us on those structures, but be sure to know what type of drug I'm going to use for what type of disorder. We have some diseases discussed in Chapter 12, so just be able to look over these two.